Good morning, everyone. Last Monday, we discussed question about individual development of the organism. Today, we will continue this lecture. Last lecture, we considered question about gametogenesis, and you must remember there are three periods of ontogenesis: pre-embryonic, embryonic, and post-embryonic period. And pre-embryonic period includes gametogenesis and fertilization. Embryonic period includes cleavage, gastrulation, primary and final organogenesis. In last lecture, we considered question about spermatogenesis, ovogenesis, about fertilization, cleavage, meroblastic and holoblastic cleavage, and gastrulation. Gastrulation is a process of formation embryonic layer, ectoderm, endoderm, and mesoderm. And we considered 4 types of early gastrulation: invagination, epiboly, delamination, and immigration, and 3 methods of formation of mesoderm. There's 3 types of late gastrulation. And next question is the first question today: neurulation. What is neurulation? Neurulation is a process of formation of neural tube. And you see on the slide neurula of amphibians. Embryo on the state neurulation is called neurula. And you see notochord, and above notochord process formation of neural tube. Neurulation begins with thickening of the ectoderm above the notochord to form the neural plate. Edges of the neural plate thicken, and you see on this slide, to form ridges. A groove forms, it depends, between ridges. The ridges fuse after that, forming a cylinder, the neural tube. The anterior end of the neural tube becomes the brain, posterior end develops into spinal cord. On this slide, you can see process of neurulation. With ectoderm, with each ectoderm above the notochord, material for notochord, forms neural plate. You see neural plate. Each mesoderm, endoderm. You can see deepening neural groove and formation of neural ridge. Blue, it's neural ridge. After that, the ridges fuse, you see, forming neural tube. Cell materials above neural tube is called neural crests. You see, blue on this slide. Each cells migrate and give rise to different structure of peripheral neural system, for example, neural ganglion. On this slide, you can see ectoderm. Ectoderm, after formation of neural tube, will called skin ectoderm. It's process differentiation of mesoderm and neural notochord and endoderm. On this slide, you see process migration cell from neural crest to other side. Next process, differentiation of mesoderm. With each electronic micrograph, you see process formation of neural groove, neural plate under notochord. With each notochord, what you see notochord pattern. Neural plate, neural groove with neural ridge fuse and formation of neural tube above notochord. And between ectoderm and endoderm, you can see process formation of it and differentiation of mesoderm. And you see body segmentation develops during neurulation. Blocks of mesoderm called somites form on both sides of the neural tube. And you see this region. Somites produce cells that form the vertebra, ribs, and muscles of the trunk and limbs. They also guide the organization of the peripheral nerves. And about neural crest. When the neural tube closes, cells called neural crest cell break loose. They migrate inward between the epidermis and the somites and under the somites. The neural crest cells give rise to many structures and organs of the body, for example, nerve ganglia, adrenal medulla. In this slide, you see process formation of neural tube again. What light blue, it's neural crests, and with both sides of neural tube and notochord, you can see differentiation of mesoderm. Each somites. On this slide you see slide embryo of chicken. On this slide you can see next structure, but this layer is number 7, it's ectoderm. Number 8 is endoderm. You can see notochord number 2 and neural tube above notochord number 1. On this side you can see better differentiation of mesoderm. Number 3 somites, number 4 intermediate part between somites and splanchnatum, number 5 number 6 splanchnatum, and number 9 secondary cavity of the body. And you can see that in the dorsal part of the mesoderm is divided into somites, and you see number 3. Each somite has the following parts: sclerotom, with these parts, sclerotom, intermediate parts, meatom, and these parts, dermatom. Sclerotom will give rise to the bones of the skeleton. Meatom is the basis for development of skeletal muscle. Dermatom gives rise to the dermis of the skin. Number 4 intermediate parts, it's nephrogonatom. These cells will give rise to gonads and kidneys. In the ventral part, the mesoderm is divided into parietal number 5 and ventral number 6 sheets of splanchnatum. You can see the secondary cavity of the body between ventral sheets and parietal and ventral sheets of splanchnatum. With this slide we will consider on practical class, and you must remember this notice. Number 1 again, neural tube, 2 notochord, 3 somites, 4 nephrogonatom, 5 parietal sheet of splanchnatum, 6 ventral sheet of splanchnatum, 9 secondary cavity of the body, 8 endoderm, 7 ectoderm. It's differentiation of the mesoderm. And the last stage of embryonic period, organogenesis. Is development of organs from three primary germlines: ectoderm, endoderm, and mesoderm. And on the slide you can see different organs which forms from ectoderm, endoderm, and mesoderm. For example, ectoderm will give rise to epidermis of skin and its derivatives, for example, skin glands, nails, epithelial lining of mouth and rectum, sense receptor in epidermis, cornea and lens of eye, nervous system, adrenal medulla, tooth enamel, epithelium of pineal and pituitary glands. Endoderm will give rise to epithelial lining of digestive tract, except mouth and rectum, epithelial lining of respiratory system, liver, pancreas, thyroid, parathyroids, thymus, lining of urethra, urinary bladder, and reproductive system. And mesoderm will give rise to notochord, skeletal system, muscular system, circulatory and lymphatic system, excretory system, reproductive system, except germ cells, which start to differentiate during cleavage, dermis of skin, lining of body cavity, and adrenal cortex. And next question, extra embryonic membranes. But before, I want to show you short film from YouTube about development of frog and development of birds. Firstly, development of frog. Cleavage, formation
The fertilized ovum undergoes rapid divisions and forms a blastula. Each division normally takes about 30 minutes.
Remember, it's amphiblastula. From micromere and macromere.
The blastula develops into a gastrula.
Formation of gastrula by epibulia and process formation from neural tube. And
Neural folds develop and fuse to form the spinal cord and brain. Eventually, the embryos hatch as free-swimming tadpoles, or larvae. The tadpoles undergo metamorphosis, and tiny frogs emerge that eventually grow into adults.
And film about development of birds.
The developmental process begins soon after fertilization and accelerates once the egg is laid, as thousands of cells rapidly multiply and organize inside the protective shell.
In the early hours, you see the foundation for this organism that's coming later on being laid down. You see the front and the back and the top and the bottom and all of those things being worked out in this cascade of events.
Primary split. It's black.
Where's the head going to be? Where's the tail going to be? What's right, what's left, what's front versus back? Already these major axes are being established. And as these cells are moving and heading towards the places in the embryo where they're going to develop, they're also changing what they're going to do eventually. They're committing themselves, in most cases, irreversibly to particular functional roles.
On day 2, the bird's spinal cord, ears, and eyes take shape. Its vascular and circulatory systems are established, and its heart beats for the first time. At hour 46, the head of the embryo starts to arch into the fetal position. Throughout the second day, a network of blood vessels branches from the embryo into the yolk. They will transport vital nutrients back to the growing organism.
You gotta pay attention to the details. You have genes being switched on and off, interaction, cell communication. It's an elaborate dance. It's like a ballet taking place on a stage with thousands of cast members. All of them doing everything they're supposed to on cue, in the right order, in the right sequence, in the right time. The process itself is so amazing. It transcends anything we know.
With the groundwork of major systems and organs now in place, the next 19 days are devoted to the completion of their development.
Yolk sac with blood vessels.
So you've gone from that one cell that can become anything in 21 days. Billions of different cells doing hundreds and hundreds of different kinds of tasks in the organism. The speed, the efficiency, the elegance of the whole process. What you're seeing is a mechanism at work. Information being translated into a physical product at each step along the way. Machines doing jobs. It's absolutely incredible. Absolutely incredible.
And continues our lecture, extraembryonic membranes. There are 5 extraembryonic membranes: yolk sac, amnion, chorion, allantois, and placenta. And consider more carefully each extraembryonic membrane. And you see this extraembryonic membrane on this slide. Yolk sac, blue, and process formation of amnion and chorion. Amnion will be and chorion outer membrane. And after that, process formation of allantois. Placenta only for mammals. Extraembryonic membranes originate from the dome lice of the embryo and function in nutrition, gas exchange, and waste removal. And you see process formation of yolk sac on this slide, yellow, and process formation of amnion, blue, chorion, and what this protrusion is allantois membrane. For mammal, the yolk sac is formed by extension of the endodermal tissue of the hypoblast. Yolk is digested by the endodermal cells of the yolk sac, and the nutrients are transported through blood vessels lining the outer surface of the yolk sac. On this slide, you can see process formation of extraembryonic organ. But consider more carefully. But each embryo of chicken, for example, and yolk. I draw embryo of chicken is big, yolk is small. In nature will be yolk big, embryo small. The embryo has embryonic and extraembryonic ectoderm, but each embryonic ectoderm on site extraembryonic ectoderm. Each embryonic endoderm on site extraembryonic endoderm, and embryonic mesoderm is on site extraembryonic mesoderm. The embryonic part of the endoderm, firstly, forms the primary gut. What you see, primary gut is formed from embryonic endoderm. What you see, formation primary gut. The extraembryonic endoderm, what you see, together with extraembryonic mesoderm, ventral sheet of mesoderm, overgrow the yolk with both sides, and forms the yolk sac. Yolk sac cells secrete enzymes that make yolk substances available to the embryo. The first blood vessels and first blood cells are formed in the yolk sac mesoderm, but in this region. They deliver the nutrients of yolk to the body of the embryo, and you see in film now. For example, put number 1 yolk sac, which consists of 2 layers, endoderm and mesoderm. Endoderm, cell of endoderm, secrete enzymes that make yolk substances available to the embryo. In mesoderm, you can see process of formation of first blood cells and blood vessels. How are the amnion and chorion formed? You see on this slide. But each body between embryonic and extraembryonic ectoderm, where this signal. Firstly, on the both sides of the embryo, but in this region, the trunk folds appear in the extraembryonic ectoderm and mesoderm. She lifts, but number 2, its trunk folds. She lifts the embryo above the yolk. And you see embryo above the yolk. Number 2, trunk folds. Red, its extraembryonic endoderm. Blue, parietal sheath of mesoderm. Therefore, lateral to the trunk folds on the both sides were number 3. Amniotic folds are formed. Number 3, amniotic folds. They grow to meet each other. Surround the embryo and connect, and you see on this slide. As a result, 2 extraembryonic membranes are formed. The inner one is the amnion, number 4. The outer one is the chorion, number 5. Amnion cells secrete amniotic fluid. It protects the embryo from drying out and mechanical shock. Chorion provides protection, gas exchange, and nutrition of the embryo. Allantois is formed from endoderm and mesoderm as an outgrowth of the intestine of embryo, and you see number 6. То есть allantois consists of from endoderm and mesoderm, like yolk sac. Amnion and chorion consist of ectoderm and mesoderm. Allantois is a bag for accumulation of harmful metabolic products. Along the mesoderm of allantois, mesoderm, blood vessels of embryo grow into the chorion, forming a special system, allantochorion. And you can see blood vessels of embryo in chorion due to mesoderm of allantois. So allantois is bridge between embryo and chorion. This allows the chorion to perform the function of gas exchange and nutrition of the embryo. Again, number 1, yolk sac. Number 2, trunk folds. Number 3, amniotic folds. Number 4, amnion. 5, chorion. 6, allantois. And you see what this extraembryonic organ on this slide too. But you see chorion, its embryo, amnion, and what allantois. Нет, it's yolk sac, а allantois What's this? And on this slide too, you see embryo brown, it's amnion, number 2, number 5, allantois, number 1, chorion, and it's embryo of mammals, and you can see next extra embryonic organ, placenta, number 7. And you see what now film and process formation of yolk sac. You see blood vessels in mesoderm of yolk sac. These blood vessels deliver the nutrients of yolk to the body of the embryo. It's smallest small embryo. Not yolk sac with blood vessels. What you see, body between yolk and yolk sac, at process formation of yolk sac. A lot of blood vessels. Embryo become bigger, yolk become smaller and smaller. Chicken was born. And next question of our lecture today, ontogenesis mechanisms. The first ontogenesis mechanism, cell division. Cell division occurs throughout ontogenesis. Cell division is the basis of the growth and development of an organism. You can see cell division during gastrulation, during cleavage, during formation of organ. Next, mechanism cell migration. An example of cell migration is the migration of neural crest cell after neural tube formation. Neural crest cells migrate throughout the body and give rise to a variety of structures: nerve ganglia, adrenal medulla, parafollicular cell of the thyroid gland, and others. Next mechanism, selective adhesion, is isolation and unification of cell of one primordium from a set containing cells of different primordia. And on the slide you see Gelfretter's classical experiments. The scientist took an amphibian embryo at the gastrula stage and destroyed the intercellular contacts with special substances. All cells were mixed, and you see on the slide, and left to stand for a while. After some time, the cells of the ectoderm formed the outer layer, you see. The mesoderm, the middle layer, and endoderm, the inner layer. Conclusion. Less digestive cell lines are located on the surface of more digestive layer. Next mechanism, differentiation. Differentiation is the process of occurrence during ontogenesis of biochemical, physiological, morphological, and other differences between uniformly homogeneous cells and their associations. For example, during cleavage, you see in blastula, in blastoderm, same cell. But through sometimes you can see nervous cell, muscle cell, epithelial cell, which have different size, different shape. Reasons of differentiations. The first reason, differences in the cytoplasm of early blastomeres as a result of ooplasmic segregation. Next reason, selective activity of genes in blastomeres and cellular induction. And you see ooplasmic segregation of this slide. Sperm fuse with egg, and cytoplasm of egg has different substances, for example, green and brown, molecules of another cytoplasmic dominant. After fertilization, you can see zygote with different substances in different parts. These different parts of the cytoplasm will be located in different blastomeres. You see mitotic cell division and two blastomeres. One blastomere has one substances, other blastomeres has other substances. These substances will affect the activity of genes in cell in different ways. As a result, some genes will be active in one cell, not in this cell, and other genes in another cell. So one cell will turn into a nerve cell and the other into a muscle cell. In addition, cells produce chemicals and release them to the outside. They can also influence the activity of each other's genes. So again, 3 reasons of differentiation. difference in the cytoplasm of early blastomeres as a result of all plasmic segregation, selective activity of genes in blastomeres, and cellular induction. And state of differentiation. First stage, state of totipotency. Preservation on equal heredity of cells. The ability of each blastomere to give rise to an organism. This ability of the human blastomere persists until state of 4 blastomeres. Next stage, state of dependent differentiation, transdetermination. The possibility of development in several directions depending on the surrounding cellular material. For example, if the cells of the mesoderm, for example, of one embryo, are transferred to the ectoderm of another embryo, then they will transform into ectoderm cells. These mesoderm cells transform in ectoderm cells because around these cells a lot of ectoderm cells. Each state dependent differentiation. Next state, state of independent differentiation, development only along a certain path even when external conditions change. On this side, if I take mesoderm cells of one embryo and transfer to the ectoderm of another embryo, this cell will develop as mesoderm cell, preserving the first path, the development. And 5 mechanism, embryonic induction. Embryonic induction is the process of the influence of some germinal structures on the development of other parts of the embryo. Embryonic induction is one of the causes of cell differentiation. When one cell can influence of development another cell. And you see scientists who study this process, Hans Spemann. Classic experiment of Spemann won Nobel Prize. Spemann took piece of presumptive neural plate from early gastrula stage and transplanted to ventral region of another early gastrula. As a result, normal development. Prospective neural ectoderm becomes skin ectoderm. Similar experiment with late gastrula. Transplanted neural ectoderm becomes neural ectoderm, regardless of transplant site. It's independent differentiation. It's example dependent differentiation. And next experiment. Transplant dorsal lip, prospective notochordal mesoderm of early gastrula, from this region, to ventral region in another embryon. As a result, induced formation of secondary embryo involving both host and transplanted tissue. You see on this slide. So you see process formation of two neural tube, primary neural tube and secondary neural tube. So you see that material of this region, dorsal blastoporal lip, which contain notochord mesodermal part, and this cell mass influence on ectoderm and ectoderm form neural tube. No? And this process, dorsal lip and discarded mesoderm opposite dorsal lip, and other embryon donor mesoderm from dorsal lip, and this mesoderm under ectoderm. And this cell of mesoderm influence of ectoderm and ectoderm form neural tube. And you see section. Primary notochord with neural tube and secondary notochord from this cell mass with neural tube. Notochord will consist of cell of second embryo, a neural tube and somates from cell of first embryo. And conclusions. Some change took place between early and late gastrula that determined fate of cells. Dorsal lip responsible for inducing shift in direction of differentiation of host tissue. Dorsal lip plays a central role in determining the craniocaudal axis of the embryo. And dorsal lip is called primary organizer. So we considered topic about ontogenesis. We considered gametogenesis, fertilization, cleavage, gastrulation, primary and final organogenesis. With this material, we will consider during practical class, and on this week or next week will be control test about question about ontogenesis. Today our lecture is over. Thank you for attention. See you later.